one of the, well, maybe I should start then by asking a question for Russell Foster from Maria, about what are the possible, what are the potential off-target effects of these molecules that might modulate sleep-wake cycles? I mean, like all drugs, they could have side effects. What sort of other off-target effects might there be? What biological pathways?
Yeah, I think it's a really good point, Maria. And of course, that's what a lot of the preclinical work we're doing at the moment is trying to resolve. What we do know is in the 4 compounds I talked about, where we've either eliminated or mutated a gene that encodes for that pathway, we don't see any obvious changes. So CT-1500 is an adenosine A1, A2A antagonist. And we've done now massive work on dose response curves and all the rest of it. And we're not seeing a problem. You're quite right. You know, all drugs have off-target effects. And that's what's going to be so important about the first in human trials, which will start in later this year for CT-1500. And of course, we would never, this needs to be resolved before we go into clinical trials. But so far, I'm immensely optimistic.
Okay, there's two questions. Oh, sorry, Maria.
Sorry, just following on from that, I just wanted to ask Russell, when we're seeing our aniridia patients in clinic, what kind of sleep screening questions should we particularly be asking?
Well, of course, I've been working closely with Susie Downs in Oxford, and we've been doing a lot of questionnaire-based sort of interviews with the anophthalmic individuals. And so there's some pretty standard questionnaires that we send over to you about screening. And in fact, one of the questions I had for you was, were you able to screen sleep-wake problems in your studies with metabolic abnormalities? It would be so interesting to see if the sleep-wake problems severity correlated with changes in metabolism, which is what you might predict.
Yeah, so we didn't, and having heard your talk and knowing the results of mine and seeing the links with, there's a lot of echo. Is that from me?
I'm getting it too.
I think it may be worthwhile anybody not speaking to mute.
Okay, yeah, that's much better. Okay, no, absolutely, and we will go back to our patients that were involved in that metabolomic study and give them those sleep questionnaires, because the link with serotonin and some of the other metabolites definitely may play a role in that. So yeah, that's really great. And just to let everyone on Aniridia Network know that we did send out an invitation for them to join your sleep study as well. So please, I would like to encourage every patient who's on this call to participate in research, because the information you give us really does inform our knowledge and how we pursue the research to lead to therapies for you.
Yeah, I think it's really important. And in view of the presentation we had on the individual effects of melatonin, I think we really, and you and I have talked about this, Maria, I think it would be great to get a proper melatonin aniridia trial to find out some solid data. There's a hint, but I think it would be great to have a proper trial on this.
It was interesting to see, though, that, and in the chat too, several people said that melatonin did work for them, and I think it might be interesting to follow that up, because maybe it's a bit different when there's a real absence of the pineal. In the case of one child who I know is part of Aniridia Network, she has been shown by MRI to have no pineal.
Yeah, I think there's a lot of individual variability. The interesting data on quadriplegic individuals who produce no melatonin at all, they show disrupted sleep, but it's no worse than paraplegic individuals who are producing normal levels of melatonin. So I think there's going to be variability, and I think what melatonin is good for is inducing sleep, and it can reduce sleep latency, and also there's about a 30-minute increase in total sleep time. So it's having an effect, but it would be fascinating to get some quantitative data in aniridia.
Thank you very much. I don't know if there are more questions for Russell, because I've got something from Elizabeth Simpson in Canada for Guy Rutter. And she says she lost sound during part of Olivia's recording. She's a long way away in Vancouver. And she says, is in a typical nonsense mutation of PAX6, would the long non-coding RNA be affected?
That's a really good question, and it would of course depend whereabouts that mutation occurred. There isn't much overlap between, I don't know if Livia showed that slide, I can't remember. There isn't much overlap between the coding regions of the two, so this would probably have to be a mutation which fell in an intron, i.e. between the coding parts of the PAX6 gene. So I think probably the short answer to your question is probably not, i.e. the coding mutations which affect PAX6 probably wouldn't influence PAX6OS1. I'll stand by in case Livia wants to correct that view.
How much interaction is there between the antisense transcript and the sense transcript? Because there is probably nonsense-mediated decay for quite a lot of the stop codon mutations.
Yeah, I mean, that's a terrific question. That's one of the first things we tried to answer when we knocked down or overexpressed PAX6OS or AS1. And frankly, it depends on the system, whether we use human cell lines or we do this in mice, we get different results, or we use primary human islets. So frankly, we don't have a consistent answer. Our view is that there isn't much direct effect of PAX6AS1 on PAX6 expression in most cases, and we think rather that PAX6AS affects more directly target genes, things like insulin and GLUT2 and so forth, which Livia described. So we don't think that in most cases PAX6AS is affecting PAX6 expression directly.
Good. Maria, go on.
I have a question for Guy, if that's okay. Guy, I think the work that Livia presented was brilliant. This is just a question more from a clinical perspective. There's a couple. So, have you actually got aniridia patients with diabetes under your care? In sort of large type 2 diabetic cohorts, has anyone ever identified any alleles, like risk alleles in PAX6? So SNPs potentially. I don't know if there are some that predisposes them to type 2. And then the last question is also for Tony, relating again to diabetes and PAX6. What do you think, if you've seen any PAX6 aniridia patients with diabetes, how would it affect the macula in terms of, like, for example, diabetic maculopathy, etc., when you have foveal hyperplasia?
Shall I answer the first two of your questions, Ben? So my clinical colleagues, I mean, we have some fairly large cohorts of people with variants of unknown impact. This is a study called the My Diabetes Study, which is led by Shivani Misra at St Mary's. None of those, as far as I know, in none of those have we detected mutations in PAX6. So we don't see a lot of aniridia patients, of course. This isn't something we do very much of at Imperial. But so the short answer is no, I haven't got any under my care or the care of my clinical colleagues. In terms of risk alleles, are they causes of more garden variety type 2 diabetes? There is a little, I'm not sure if it's a very common cause. PAX6 wasn't one of those genes which popped up in GWAS for type 2 diabetes, as far as I recall. So I don't think there are common variants in the PAX6 gene which seem to drive type 2 diabetes risk.
I had heard Anna Gloin say about a year and a half ago that they did find a signal at PAX6. But I have been looking out for a paper and I haven't seen it yet. I've also heard from Maria, it's the woman that I put you in touch with in Cambridge. I forgot.
Yes, yeah. Sadaf Farooq.
Sadaf Farooq.
Sadaf Farooqi, who works with, in Stephen O'Reilly's unit.
Just before I wrote to her to put Maria in touch with her, she had been looking at some non-coding variants in the PAX6 area of interest to them. But that's all the information I have. I mean, I'm a great believer in lots of non-coding variants having effects on gene expression, and therefore it's just possible.
Yeah, I think the
latest GWAS for type 2 diabetes have reported something like 400 signals, but the more that one does, and with huge numbers of patients and 100,000 sort of subjects and 1 million controls and so forth, you will begin to detect signals with very, very low effects. So there may be a signal close to PAX6. And by the way, even if you see a variant that's close to the gene, you don't know that it's actually PAX6 that's the causal transcript.
No, well, the PAX6 region has been quite widely analyzed. Kelly, I was very interested to see the possible link between PAX6 and PAX2. I think that's really interesting. I wonder, is anybody following that up?
Not yet.
No, I mean, it's well established that they are interacting with each other and they co-bind to targets and so on. But I didn't ever think that they might be implicated in WAGR.
It is a fascinating and I think could be a very productive line of investigation. So as I mentioned, our registry data is open to any investigator. So we would welcome additional collaborators.
Ngozi? I can't hear you.
I think you're muted, Ngozi.
You need to unmute.
There is no muted button next to her. It's not working. While we're waiting, because we only have a...
Tony, could you just...
Yeah, I said I'd... Yeah. The other... I want to make one point about the diabetes and aniridia. And that is, when I was just sort of looking up, last time I read the papers on it, people have... there's a number of papers on abnormal glucose tolerance tests in patients with aniridia, small cohort studies. But one of the issues, and it's the same issue with your metabolomics in a way, when Sanjay Sisodia was doing the original work on, and he's done some other work on aniridic patients and various abnormalities, cognitive tests and other things, you actually need visually impaired controls. Because if you're visually impaired, your diet changes, you are more likely to have a high BMI, and it's very important to use the correct controls. And this was brought home because when John Yates and I, many years ago, started our GWAS study and genetic epidemiology study of AMD, we spoke to the MRC nutrition unit about diet questionnaires and storing various metabolites in various samples. And they thought it would be of no value because they said when you become blind, your diet changes quite significantly. And so I don't think there's good enough studies yet to show that type 2 diabetes is more prevalent in aniridics. I mean, I know your study showed quite a high prevalence, but we need probably better controls. In terms of the second point about foveal hyperplasia and what effect that has in later life. The answer is it would be pure speculation. I know from helping someone recently to write a sort of an MRC grant application who's working on foveal development. I said that question might be asked by the panel, and if you look at the big, there is data from the big study of what's the name of it, Maria? The big study of
100,000.
Yeah. No, yeah. Where they've got OCTs of everybody. I don't think that's particularly helpful. So the answer is I think it's unknown. Although it's very interesting that our colleagues who work on treatment of AMD had said to me once they'd never seen AMD in a patient with albinism, and they live a long time. But so there may be something in it, but it's pure speculation.